and human welfare. What we need to go for in the new world is not the application of physical science for production of goods to get money power, but application of biological science to build better men and a better society. The first step in the new world must be the abolition of poverty, and we must concentrate on building men and women before we build new cities. The age which is now passing away is largely the age of physical science, with its invention and discoveries which have given us power over the forces of nature. It is to be hoped that when this war is over, the age of physical science will be replaced by an age of biological science, the study of life in all its manifestations. Biology is the contributing in many aspects of human life, for instance, human population is increasing continuously. Approximately 7 billion people are living on the planet Earth. All of our food and many daily usable home articles are derived from either plants or animals. The development of improved high-yielding varieties of these organisms is possible through the biological research. Due to the modern invention, the consumption of energy has been increased manifold. Therefore, many fourth-generation plants have been installed and are being installed. These plants and the use of modern invention in our daily life also drastically change integrated disease management. Effective control of particular disastrous disease of all the common diseases of a population can be achieved by using all relevant appropriate methods of disease control. Such an approach of disease control is known as integrated disease management, combating of disease by utilizing all methods as and when required and ensuring the participation of community in this program is very useful. Way of these control thus requires an awareness of the community about severity of the problem, its causes and its remedies. Public awareness can be ensured by using print and electronic media, by arranging seminars in schools and colleges, or by person to person communication. In integrated disease management, every available method of disease control is used like preventive measures, drug treatment, vaccination, and different kinds of therapies. Actually, the real objective is to stop the further spread of disease and to prevent its new onset. This has proved very effective program for elimination and control of the dangerous disease from the human society. Vaccination. Vaccination is the administration of vaccine stimulate immune system of an individual to develop artificially induced active immunity against an infectious disease. Vaccines can prevent the effects of infection by many pathogens. A vaccine may be intact but inactivated, non-effective, or attenuated with reduced infectivity. Form of the causative pathogens, bacteria or viruses, are purified components of the pathogen that have been found to be highly immunogenic. For example, the outer coat proteins of virus particles. Toxoids are produced from further immunization against toxin-based diseases such as tetanus plasmine of tetanus by destroying the toxic but preserving the immunogenic effect. How does vaccine work? When the body is exposed to the weak or dead organism vaccine, the body is integrated to produce antibodies. Since the injected agents are weak or dead, the body does not actually suffer the disease, but an immune response is initiated. Now the body is fully equipped to fight against the actual causative agent like virus or bacteria that attack the body later in life. The same principle work in the body in case of natural active immunity. This is the reason why some childhood diseases occur only once in the lifetime. Main important point. History of vaccine. Edward Jenner was the first scientist who developed a vaccine against smallpox in 1796. He conducted an experiment on an 8-year-old boy, James Philip. He scratched the skin of the boy and introduced into the area of a liquid of cowpox obtained from the hand of milkmaid who contracted it from cow. Later, when boy was exposed to smallpox, he did not suffer the disease. James Philip thus immunized against smallpox. Later, Louis Pasteur discovered that aging or attenuated culture of bacteria that cause all cholera when introduced into healthy chickens developed immunity against that disease instead of causing the disease. Pasteur next applied this principle of inoculation with attenuated cultures to the prevention of anthrax and rabies. And again, at work, the term vaccine was proposed by Pasteur, who honored the Edward Jenner because the term is derived from the Latin vacca, which means cow, because the first vaccine was made from cowpox liquid. Types of vaccines. Scientists take many approaches to designing vaccines against a microbe. These choices are typically based on fundamental information about the microbe, such as how it infects cells and how the immune system responds to it, as well as practical considerations such as regions of the world where the vaccine would be used. The following are some important types of vaccines: lab attenuated vaccines, inactivated vaccines, subunit vaccines, toxide vaccines. Live attenuated vaccines. Live attenuated vaccine contain a version of the living microbe that has been weakened in the lab so it can't cause disease because a live attenuated vaccine is the closest thing to a natural infection. These vaccines are good teachers of immune system that elicit strong cellular and antibody responses and often confer lifelong immunity with only one or two doses. Despite the advantage of live attenuated vaccine, there are some downsides. It is the nature of living thing to change or mutate, and the organism used in live attenuated vaccines are no different. The remote possibility exists that an attenuated microbe in all vaccine could revert to a virulent form and cause disease. Examples include the viral diseases yellow fever, measles, rubella, and mumps, and the bacterial disease typhoid. Inactivated or killed vaccines. Scientists produce inactivated vaccines by killing the disease causing microbe with chemical, heat, or radiation. Such vaccines are more stable and safer than live vaccines. The dead microbes cannot mutate back to their disease causing state. Inactivated vaccines usually don't require refrigeration and they can be easily stored and transported in a freeze dried form, which makes them accessible to people in developing countries. Most inactivated vaccines, however, stimulate a weaker immune system response than do live vaccines. So, it would likely take several additional doses or booster shots to maintain a person's immunity. Examples are influenza vaccines, cholera vaccine, bubonic plague vaccine, polio vaccine, hepatitis vaccine, and rabies vaccine. Subunit vaccine. Instead of the entire microbe, subunit vaccine include only the antigens that best stimulate the immune system. In some cases, these vaccines are epitopes, the very specific part of the antigen that antibodies or T cells recognize and bind to because subunit vaccine contain only the essential antigens and not all the other molecules that make up the microbe. The chances of adverse reaction to the vaccine are lower. Example include the subunit vaccine against hepatitis B virus that is composed of only the surface proteins of the virus. Toxide vaccines. For bacteria that secrete toxins or harmful chemicals, a toxide vaccine might be the answer. These vaccines are used when a bacterial toxin as the main cause of illness. Scientists have found that they can inactivate toxins by treating them with formalin, a solution of formaldehyde, and sterilized water. Such detoxified toxins called toxides and are safe for use in vaccines. Vaccines against diphtheria and tetanus are examples of toxide vaccines. Vaccination against some common diseases is given. Polio, OPV, oral polio vaccine, live vaccine from birth to 5 years of age. Tuberculosis, BCG, Bacillus calmette-guerin, live vaccine at birth. Typhoid, typhoid vaccine, TB vaccine, typhoid para, typhoid A, paratyphoid B, killed vaccine at 2 years of age. Hepatitis B vaccine, subunit vaccine at any age. Diphtheria, tetanus, diphtheria toxide vaccine, tetanus toxide vaccine generally in childhood. Animal husbandry. Ever since the beginning of civilization, humans have depended on animals for many requirements such as that of food, milk, meat, and egg, clothing, hide or wool, labor, pulling, carrying, load, security, etc. The development of desirable qualities in all such animal species through creating better breeds has been an important human achievement. For this, humans have consistently tried to improve the breeds of domesticated animals to make them more useful for them. The branch of science which deals with the study of various breeds of domesticated animals and their management for obtaining better products and services from them is known as animal husbandry. The term husbandry derives from the word husband, which means one who takes care. When it incorporates the study of proper utilization of economically important domestic animals, it is called livestock management. Being a country that has largely agriculture-based industrial system, animal husbandry plays an important role in the rural economy of Pakistan and as a major source of livelihood for many farmers. It is estimated that there are between 30 to 35 million people in Pakistan current labor force who are engaged in livestock. Different categories of animal livestock. Wild, those that breed better when they are free than they do when they are captivated. They have no common use for humans. Examples are lion, tiger, rhinoceros, deer, etc. Then, those who are caught from the wild and trained to be useful to humans in some way. Elephant, chimpanzee, gorilla, yak, etc. Domesticated. Those that are of use at home and are easily bred and looked after by humans. Common domesticated animals are dog, horse, cow, sheep, buffalo, fowl, etc. Comparison of average milk yield across countries show that one New Zealand dairy animal produces as much milk as three dairy animals in Pakistan, while one American cow produces as much as seven Pakistani cows. Importance of domesticated animals. On the basis of utility, domesticated animals are categorized into the following functional groups: milk yielding animals, cattle, buffalo, goat, sheep, etc. Dirt animal used for load, bullock, horse, donkey, mule, bearing camel, elephant, yak, etc. Fiber, hide and skin yielding sheep, goat, cattle, buffalo, camel, etc. Meat and egg yielding animals, fowl, hen, duck, goat, buffalo, etc. Types of husbandry. There are several forms of animal husbandry. Out of them, some important forms are given below. Milk and meat yielding animals. Depending upon the availability and regional consideration, different animals are reared for the purposes of yielding milk and meat. Cattle are considered to have been one of the first animals domesticated by man for agricultural purposes. Cattle mainly include cow, bull, oxen, goat, sheep, etc. The females of the species provide milk, which in turn contribute animal protein to the diet of people, while the female species of these cattle are used for milk. The male species play an important role in agricultural economy by providing labor, meat, and hide. Milk itself is taken in many forms like ghee, curd, butter, cheese, etc. The excreta of these animals used as manure and biogas and as fuel. Dairy products. Milk is drawn from the animal as known as full cream milk. When the cream is separated and remaining milk is called toned milk. This milk contains no fat and is known as skimmed milk. On the basis of fat contents, the various milk products are as follows: cream. It is prepared by churning milk. The fat comes on the top, which is separated by draining out the liquid. It is known as cream with 10 to 70% fat contents. Curd. Milk is converted to curd due to bacterial activities. Buttermilk. It is the leftover liquid after removal of butter, ghee. After heating butter, the water evaporates and fat content are almost 100%. Condensed milk. Milk is concentrated by removing water contents with or without adding sugar. It is 31% milk solids with 9% fats. Powdered milk. It is the powdered form of milk cheese. It is coagulated with protein, casein with fat and water. Koya, a desiccated milk product prepared by evaporating water contents and reducing the bulk to about 7 to 75%. Cattle dung. Cattle dung is mainly used to make dung cakes for burning as fuels. It is used mainly in villages. The farmers also use cattle dung to produce biogas and the leftover residue as manure. Biogas plant gobar, gas plant, biogas plant as chamber where animal excreta, cow dung, buffalo dung, etc. and some anaerobic bacteria are put into a tight biogas chamber. Decomposition of excreta produces methane gas used as a smoke-free gas for cooking. This gas can also be utilized for lighting. The leftover solid residue serves as good manure. Draught animals. Draught animals are animals neat for carrying load. From ancient time, a number of animal species have been used for special purposes by humans utilizing their mechanical strength, endurance, and speed. These include horse for riding and swift running, elephant for riding, strength and heavy load lifting, camel for riding and sandy desert and ability to survive without water for long duration, donkey and mule, a hybrid of male donkey and female horse for carrying loads. Most of the drought animals are herbivorous and survive on leaves of trees, shrubs, and bushes. While raising them, they are also fed on grains, beans, cotton seeds, maize, and bran beside dry green fodder. Fiber, hide, and skin yielding animals. Besides providing meat, milk, and transport, livestock provide many commercially useful products such as fiber, skin, and hide. Generally, sheep and goat provide fibers for making of products like woolen strings, roofs, carpets, clothing, and brushes, etc. Egg yielding animals. This category consists of egg producing animals whose eggs are used as food by mankind to provide proteins. Poultry farming is defined as term for rearing and keeping of birds such as fowl, duck, and hen for egg or meat. Poultry farming has become popular because it is comparatively easy to start and maintain. It gives quick return within 1 to 6 months of investments. It is easily manageable and requires less space and labor. Poultry birds and their eggs are a rich source of nutrients. Genetic improvement in animals. The application of laws of animals' health and reproduction genetics has contributed toward increase in milk, eggs, and meat productivity. The increase in egg production brought about the silver revolution in the area of animal husbandry. The methods being widely used are artificial insemination and embryo transplant. Artificial insemination. Artificial insemination involves collection of semen from a healthy bull of the desired breed, its storage at low temperature, and introduction into the females of cattle of other breeds for bringing about fertilization using sterilized, germ-free equipment. Advantages of this method are up to 3,000 female can be fertilized from semen collected from one bull. The semen can be stored for a long period and transported over long distances. It is economical and had high success rates of fertilization. Embryo transplant. This method of breed improvement has been quite successful in sheep and goat. In this method, embryos, depending on their period of development, from superior breeds are removed during the early stages of pregnancy and are transferred to the other female with inferior character and whose body the gestation period is completed. By this technique, quality and productivity in the livestock can be improved. Unlike artificial insemination, this method has low success rate due to greater chances of contamination. Latest techniques used for plants. From ancient times, plant breeders have been struggling for the improvement of crop plants that breeders have tried to incorporate into crop plants by using various techniques in the last 100 years include increased quality and yield of the crop, increased tolerance of environmental pressure, salinity, extreme temperature, drought, resistance to viruses, fungi, bacteria, increased to tolerance to insect pests, increased tolerance of herbicides. Classical breeding relies largely on homologous recombination between chromosomes to generate genetic diversity. The classical plant breeder may also make use of number of in vitro techniques such as protoplast fusion, embryo rescue, or mutagenesis to generate diversity and produce hybrid plants that would not exist in nature. In addition, many new techniques are also being employed by breeders for improvement of crop plants. These techniques may include acclimatization, selective breeding, hybridization, and back crossing. Plant improvement by genetic engineering is also becoming popular day by day. Acclimation is a process of an individual organism adjusting to a gradual changes in its environment, such as a change in temperature, humidity, photoperiod, or pH, allowing it to maintain performance across a range of environmental conditions. Acclimatization occurs in a short period of time, days to week, and within the organism lifetime compared to adaptation. And acclimatization are different terms in the sense of acclimation is used under laboratory conditions while acclimatization is in the field or in nature. In order to maintain performance across a range of environmental conditions, there are several strategies organisms use to acclimate. In response to changes in temperature, organisms can change the biochemistry of cell membranes, making them more fluid in cold temperatures and less fluid in warm temperature by increasing the number of membrane proteins. Organisms may also express specific proteins called heat shock proteins that may act as molecular chaperones, protein that assists non-covalent folding and help the cell maintain function under period of extreme stress. It has been shown that organisms which are acclimated to high or low temperatures display relatively high resting levels of heat shock proteins so that when they are exposed to even more extreme temperatures, the proteins are readily available. Expression of heat shock protein and regulation of membrane fluidity are just two of many biochemical methods organisms use to acclimate to novel environments. Many plants such as maple trees, irises, and tomatoes can survive freezing temperature if the temperature gradually drops lower and lower each night over a period of days or weeks. The same drop might kill them if it occurred suddenly. Studies have shown that tomato plants that were acclimated to high temperature over several days were more efficient at photosynthesis at relatively high temperature than were plants that were not allowed to acclimate. Selective breeding. Selective breeding is the art and science of changing the genetics of plants in order to produce desired characteristics. Plant breeding can be accomplished through many different techniques, ranging from simply selecting plants with desirable characteristics for propagation to more complex molecular techniques. Sometimes many different genes can influence a desirable trait in plant breeding, the use of tools such as molecular markers or DNA fingerprinting can map thousands of genes. This allows plant breeders to screen large populations of plants for those that possesses the trait of interest. The screening is based on the presence or absence of certain genes as determined by laboratory procedures rather than on the visual identification of the express trait in the plant. International development agencies believe that breeding new crop is important for assuring food security by developing new varieties that are higher. high yielding, resistant to pests and diseases, drought resistant, are regionally adapted to different environment and growing conditions, hybridization and back crossing. Classical plants breeding uses deliberate interbreeding crossing of loosely or distantly related individuals to produce new crop varieties aligned with desirable properties. Role of genetic engineering and crop improvement. Since last 2 decades, genetic engineering has been imparting a very significant role in crop improvement. Desired traits gene can be inserted in the plant genome by genetic recombination using the bacteria, Agrobacterium tunificans, or rhizogenes, or by direct method like the gene gun microinjection. The, the majority of commercially released transgenic plants are currently limited to plants that have introduced resistance to insect pests and herbicides. Insect resistance is achieved through incorporation of a gene from Bacillus thuringiensis (Bt) that encodes a protein that is toxic to some insects. For example, the cotton bollworm, a common cotton pest, feed on Bt cotton. It will ingest the toxin and diherbicide usually work by binding to certain plants' enzymes and inhibiting their action. The enzymes that the herbicide inhibit are known as herbicide target sites. Herbicide resistance can be engineered into crops by expressing a version of target site proteins that is not inhibited by the herbicide. This is method used to produce glyphosate resistant crop plants. Home gardening. Home gardening is popular hobby for number of reasons such people garden for exercise and some for enjoyment of harvesting. Flavorful produce that can be eaten fresh from the garden or preserved for use during winter. Other garden so they can grow a wider variety of vegetables and herbs than are available at their local grocery store. In recent years, many people are growing vegetables to save money on their grocery bill. Flower gardens are planted as a hobby and their aesthetic beauty. A garden can be a wonderful place for children. They provide opportunities for play, learning, and for having fun. Increased. So has the need for increased. So has the need for more information and education. Careful planning can make gardening easier, more productive, and more enjoyable. In planning your garden, it is important to consider a few basics. Your garden, it is important to consider a few basics. Many environmental plants files the home